The phrase science journalist sort of sounds kind of techy and maybe a little bit, you know, clinical, and it doesn't capture the empathy of this woman. The topics she chooses to focus on and the kinds of questions she asks and the way she conveys what she's learned is really extraordinary, and this book is an example of that, The Angel and the Assassin.
So microglia are both angels and assassins. One of the nice things that they do is when they're in homeostasis, like our immune cells in the body, they run around being protective. They're the angels of the brain. So they run around and they cater to the needs of neurons, kind of the way an entourage caters to the needs of a movie star. They make sure that neurons are happy, they're bathed in these groovy growth factors and protective chemicals. One of the other things they do, as best I understand it, is they also help neurons to make these protrusions so that they can fire and wire together. And when we say that they engulf, microglia engulf synapses, we mean that they're taking pieces of the synaptic material and they are eating pieces of the synaptic material when they should be helping the synaptic material to form these protrusions and fire and wire together. And this was the question that intrigued Beth Stevens most. If microglia, which researchers now understood, were akin to the white blood cells of the brain, and they were altering brain circuitry, perhaps microglia didn't always get it right. What if, instead of just pruning away damaged or old neurons, microglia were sometimes mistakenly engulfing and destroying healthy brain synapses too? Increasingly, disorders of the brain, from depression to learning disorders, were being understood as circuitry disorders. Certain synaptic connections in the brain were not firing and wiring together as they should. Beth explains, what if these little overlooked glial cells we were setting out to study were at the center of it all? Her hands spread wide with excitement. You can imagine how, if you have too much pruning or too little pruning, things go awry. You could have too many synapses or not enough synaptic connectivity. And you can imagine, given how our brain works, if that connectivity is even slightly off, that could potentially underlie a range of neuropsychiatric and neurodevelopmental disorders, as well as cognitive impairment. Could it be that sometimes microglia were the brain's sculptors and protectors, the angels of the brain, and sometimes they were the brain's untimely assassins? Was it possible that scientists had, for so many decades, missed something so huge? No one knew. If Beth could prove this grand, complex hypothesis, it would completely change our understanding of brain health from womb to grave. But in order to prove her hypothesis, she first had to get a better look at these little cells. Beth hypothesized that if indeed microglia were engulfing and destroying synapses, then she would see chewed up synapses inside the microglia. That was the big question, Beth explains. Could we find pieces of synapses inside the microglia? Dory, my postdoc, figured out a really clever way to allow us to see if that was indeed the case. Dory Schaefer, now an assistant professor of neurobiology at the University of Massachusetts Medical School, explains their experiment, one that is now universally considered a landmark study in the field of neuroscience. In order to get a closer look at how microglia were interacting with synapses, Dory injected dye into the eyes of mice. This dye then traced down from the neurons and the eye nerves and deep into the brain. This, says Dory, made the brain's synapses glow a bright fluorescent red. Microglia, on the other hand, were stained fluorescent green. And this, Dory says, allowed us to clearly see both the synapses and the microglia, since each glowed a distinct fluorescent color. This process, simply finding a way to accurately see the interplay of synapses and microglia inside the brain, took nearly a year. Dory recalls, one weekend I was in the lab by myself, imaging and re-imaging microglia and synapses. I'd looked through the microscope a million times. Then suddenly I saw these little red structures, synapses glowing like fluorescent red lit up dots. And these red dots were inside the belly of the green microglia. Dory was stunned. I kept thinking, we were right. Microglia are eating synapses. I was staring at proof positive with my own eyes. The synapses were inside the microglia, and microglia were chomping them to pieces. Beth recalls Dory running into her office that Monday morning with photo images in her hand. They're in there, Dory told Beth. The synapses are really inside the microglia. We can see it. It was such a high-five moment, Beth recalls. Microglia were like tiny little Pac-Men in the brain, and brain synapses were in the bellies of the Pac-Men. We were on to something really wonderful, really novel. This was deeply important in terms of looking ahead to microglia's role in disease. Beth and Dory rolled up their sleeves. We were both really fired up, but it was also really hard and stressful, Beth recalls. She was pregnant with her daughter Zoe and had a toddler at home. Dory and I knew we had to crank. We had so much adrenaline, but we also wanted to be rigorous and take our time doing our control experiments and data analysis. We wanted to do it right and publish it before anyone else. It was a tall order. The following weeks and months were reminiscent of Beth's early days as a scientist, back when she'd started at NIH. Many nights she'd look up at the clock and it would be close to midnight. It just wouldn't make sense to go home, she says. But one thing had changed compared to her early days as a lab tech. Beth no longer had to sleep on a pile of clothes under the lab conference table. Laughing, she tells me, Dory gave me a blow-up mattress as a present. I put it under my desk, and if it was really late, I'd crash there. In 2001, Beth and Dory submitted a paper outlining their findings for publication and review. Beth had recently given birth to her second daughter, Zoe, and now had 2 young children at home. And Dory had recently gotten married. It was a watershed year. In 2012, their seminal paper was published in the journal Neuron. It was the first scientific study offering evidence that microglia were pruning and eating away synapses. Not surprisingly, the scientific world exploded with the news. Their study was later cited as the journal's most influential paper that year. Meanwhile, researchers at the European Molecular Biology Laboratory in Italy showed that microglia could be especially overactive in the brain's hippocampus, an area crucial to mood and memory. By engulfing and removing healthy synapses in the hippocampus, microglia were causing a loss of brain matter in a part of the brain that was known to be deeply affected in depression, anxiety disorders, obsessive-compulsive disorder, and Alzheimer's disease. In these diseases, the brain's hippocampus had been shown through PET scan imagery to be visibly atrophied. These revelations solved a decades-old mystery. In many different neuropsychiatric and neurodegenerative diseases of the brain, healthy synapses disappeared, but no one had ever understood why. Suddenly, it all made complete sense. Even though microglia were trying to protect the brain and make it healthy and strong, just like white blood cells in the body, when microglia detected that something was off, an overinflux of stress hormones, an infiltrating virus, chemical, or pathogen on the scene, they all too often went too far, removing every synapse hanging out in the neighborhood. And that understanding that this little boring cell that had been hiding in plain sight was an immensely active and sometimes hyperactive immune cell changed everything. Well, so in healthy development, microglia have a really good role, and during development, you have way too many synapses and they just keep growing. And if they kept growing like that, you know, it'd be kind of like the limbs of a tree. You'd have so many that the tree would just fall over. It wouldn't be healthy. So some of those synapses have to be pruned back. And there is that good role for microglia to prune back synapses for healthy development. But later in life, in adolescence with mood disorders or years before the symptoms of Alzheimer's even set in, if that pruning begins a runaway process, then we begin to have a problem. We begin to see symptoms of depression or anxiety or a loss of synapses, which is really something that happens years before we see symptoms of Alzheimer's. You know, one of the ways in which we can look at that is through the science of looking at the connectome in the brain, which is just a fancy word for the way in which different areas of the brain, important areas of the brain, like your amygdala, which is your alert center, it tells you to go into fight, flight, freeze, or your hippocampus, which helps you store all of your memories or process what's real and what's not real, or your prefrontal cortex, which is helping you make decisions. All of those areas of the brain, they have to talk to each other, they have to communicate, they have to phone each other all the time. And in order to do that, all those phone lines have to be working perfectly. But if over time, some of those connections begin to go down, and that process continues to unfold, then we begin to have something called runaway neuroinflammation. And that simply means that microglia are caught in this process of continuing to prune the brain. Think of it as a slow brew process over time in a way that we may not see symptoms immediately, but eventually symptoms will come. I think that is a two-part answer. On the one hand, this science, to me, is the most profound, most complex, admittedly, but most profoundly hopeful science I've ever reported on. I've written quite a few books, because as we have this new understanding of the brain, we also have this race to understand and see signals for when microglia begin to shift from, remember we talked about how they're catering to neurons like movie stars, they're bathing neurons in synapses and protective factors. Well, this work at Broad Institute in Boston and many other places is starting to assign what we could think of loosely as barcodes to every state that microglia can be in. So your happy microglia are running around and they're taking care of your synapses, and you're thinking pretty well and you're feeling pretty great. As they begin to shift and become assassins, and they start to spit out these inflammatory factors, or they start to morph into these big bushy Pac-Men, they literally change their shape and size. If we can assign different kind of like supermarket barcodes to them as they begin to change their activity and test for that, we can begin to know when they're starting to change their actions. That sounds like crazy far off science. It's not, it's happening. It's not here right now, but some aspects of it are here right now. At Shock Trauma, at STAR Research over at Shock Trauma, the research arm of Shock Trauma, they've been able to, Alan Faden has been able to look at factors that are released by microglia in the brain. These factors descend into the bloodstream and can be measured in blood tests. And this is being used now in concussion. So if somebody has a concussion, it may look like a mild concussion, right? Or it's hard to tell who is responsible in the brain. What's responsible for the severity of concussion? Big surprise, microglia, right? And so as microglia get active after a concussion, how active they become determines whether that concussion is mild or severe or anything in between. So if we can look at these descendants of microglial activities, these factors that they express, and go, just like you might take a white blood cell count, right? Everybody knows you think you're sick, you go to the doctor takes a white blood cell count. If we can begin to look at their activity and know that somebody's concussion is really severe, then we can treat it differently. A doctor can ask that patient to do things very differently than they might for a mild concussion. And guess what else we can do as the science comes closer to reality? We can determine which treatments are working for people, right? So right now, if you have a patient with a concussion, early Alzheimer's, because that's it, the longer something's in motion, the harder it is to intervene. Depression, or let's say you have a 12-year-old girl in a family with a lot of depression, and maybe there's a lot of Alzheimer's in that family, and maybe that child also had a concussion at 10. We may have a time, and I don't think it's that far off, where a pediatrician might be able to suggest a blood test, kind of like we test for cholesterol, and say, there is a lot of microglial activity here. Why don't we think about ways, and this gets to the second part of the hope, that we can address the brain the same way that we're keeping your child's immune system healthy. So the second most hopeful thing to me is that we don't tend to think of the brain the way we think of our body. We don't think to protect our brain's health the way we think of our physical health, and they are one and the same. So that to me is profoundly hopeful, that you can help people to understand protecting the health of your brain's immune system is something we need to be thinking about every day and from a very early age. 100%. And I think everybody here who reads my books knows that I write secretly from a feminist perspective, right? Like, you'll notice that when I talk to Peg about scientists for this book, I wanted a female scientist. Now, it just happened that she also was the major scientist in this area. If she hadn't been, I would have had to think about that. No, I'm really serious. But women have been held back in science in so many ways, and in so many other fields. And yet, these diseases that women like Beth and Peg are studying, and my friend Christy Bethel back there from Johns Hopkins, these are kick-butt women. I admire them. I see what they're doing, and women are also the ones who suffer the most from these disorders. I call it the 3-to-1 rule, and this is kind of like fuzzying the statistics a little bit, but roughly girls are suffering from depression at 3 times the rate of boys, roughly 3 times the rate of anxiety. In fact, I saw some stats that Christy sent me last night about, in the last 10 days, how many more girls report mood issues versus boys. Women, we now know, are suffering from Alzheimer's at 3 times the rate of men. We know that after childbirth, the rate of, we have these inflection points in adolescence, after childbirth, and after menopause, during menopause, where shifts in estrogen and many other complicated things, as best I understand them, change the way our immune system responds. It shifts the female immune response into overdrive. The female immune response it gets stuck more easily on overdrive. That's just the simplest way that I can put it. And if these disorders are all happening 3 times more to women and girls than men, that doesn't mean that we don't need to talk about them happening to everyone, but we have to talk about them with the mindset and the understanding that we haven't really been researching women. So yeah, did I want to tell women's stories? Yeah. Did I want to portray the work of women scientists? I did. I can do that. It's my book. You can read the book and you'll see that there are a lot of things happening. I put them into 3 buckets. Things that are here and have kind of been around for a long time. We can see that fasting mimicking diets in moderation and fasting diets are helping our immune cells to kind of shrink and reboot. And there is a lot of excitement at Hopkins and also at USC that this is causing microglia to reboot. Now, when I say that, I want to be clear that this is the thought, this is the hypothesis, this is what they see happen. Has somebody watched it happen literally under a high-resolution microscope in real time? No, but this is where the understanding lies. Gamma, so those first buckets are things, some of them are common sense. Obviously, rigorous exercise is really good for the brain. Some things that have been around, like neurofeedback, have come back under new scrutiny because of the way in which they affect brain waves in the brain and the direct current field in the brain, which emits brain waves. This direct current field helps to establish brainwaves. And brainwaves are like what make us kind of happy or chill when we need to be. In Alzheimer's, gamma waves are really impaired. And microglial cells help to make that underlying current in the brain. So it's brought a new focus to old things like neurofeedback, which have like a 30 to 40% efficacy rate for depression, which is not that different than some antidepressants. So it's interesting that there are things that are out there that are available that are FDA approved. Then bucket 2, new things, transcranial magnetic stimulation, which is this non-invasive pulse from outside the brain, which is looking to, again, there are edges that are, things at the edges of our understanding we don't know, but look to be rebooting the activity of microglia because synapses are able to come back online. Bucket 3, not really here yet. Everybody asks me about gamma light flicker therapy. So this is time I spent at the Peak Hour Institute at MIT with Li Wei Sai, who showed me this little Tupperware box that she made with her team. She'd taken computer software and she created this wand. Literally, this was like a Tupperware box and duct tape, black duct tape. And this wand pulsed 40 hertz, LED lights at 40 hertz outside the brain in mice. Let's be clear. That's why I put it in bucket 3. It's not here. And it caused microglia to not only stop being the assassins, but to start clearing the brain of tau protein. Microglia reemerged as their angelic cells. And the results lasted in mice. We're not mice, we're humans with Alzheimer's, or in the Alzheimer's model, for up to a week. So I asked her in her office, are you doing it? Because this sounds pretty sweet, right? Like, I don't want to start forgetting. I would like my tau protein to be cleaned on a regular basis and my microglia to come back to their good angelic entourage state. Yes. And so then she said, and you'll read it in the book, I've got a half of MIT lining up outside my door. So those are in clinical trials. I know exactly where they are in clinical trials. I can't really say right now, but it's coming along. It's coming along. Ketamine, big study concluding right now. I wrote about it in an op-ed for Stat on Friday. The BioK study is coming along, and if it comes out the way, if the results are positive, researchers will be able to look at patients' levels of inflammation in the body, hypothesize from that there is neuroinflammation in the brain, microglia are spitting toxins and sculpting synapses, over-pruning, and know that patient is more likely to respond to ketamine for treatment-resistant bipolar and depression, because the theory follows, you know where I'm going, that treatment-resistant depression, bipolar, anxiety disorders are because of Runaway microglia.